Technologies. We are the authorized distributor for Nanostring technologies in Malaysia. So usually I'm the one who provides the application support, basically from pre-sales discussion, experimental design, on-site support for assay run, instrument operation, down until data analysis. So we usually start from start to end. We provide support for all our Nanostring users in the region in Malaysia. So Nanostring is actually a US-based company based in Seattle. It has more than 800 employees, and they actually started off coming out with this nCounter analysis system, which is specialized for doing multiplex gene expression assay. And up until 5 years ago, they started to move into spatial biology. So over the span of 3 years, in 2019, they came out with this Genomax digital spatial profiler. And then just 2 years ago, they come out with a even newer platform called Cosmix Spatial Molecular Imager. And in the same year, they also came out with this Atomic, which is used for it's a cloud-based data analysis platform, which helps the user to analyze their spatial biology data on cloud. So there are two different these two different instruments. Both of them are doing spatial biology, but in terms of the difference is mainly in terms of the resolution, the level that they provide. So based on your research need, different instrument here will probably be more suitable for you. So of course, today we are going to be more focused on talking about these two spatial biology platform. But at the end of the talk, I'll also talk about briefly on this nCounter analysis system as well. Because in the case that if you do not need the spatial biology and then you just want to do a photo RNA gene expression study, then nCounter is still something that you can do into. So first, probably you can, using fruits as an analogy, we can see why is it important, or why do you need to do spatial biology. So back in 2006, when either using the nCounter platform or when NGS first came out, it was possible to do bulk genomics. So basically, you extract everything from your tissue section, for example, and then you just basically profile the whole genetic expression of proteomics from the whole tissue section. So basically, this is what we call bulk genomics. So from there, you will be able to get the average expression level for your genes, and then can compare the profile in between different tissue sections or different tissue you get from different patient or different disease. Then you can compare from there whether there's any differential gene expression. So there's a lot of information can be obtained from bulk genomics, but the problem is if you use fruit as an analogy, it's basically a blend of fruit juice. So you know what you get is basically just a blend of everything. You get an average value out of everything that you have extracted from the tissue section. And then move forward to 2012. 10x Genomics came out with the system called Chromium that is able to do a single cell sequencing. So right now, other than bulk genomics, now you can actually split out the signal and then to find out whether this the relative abundance of different cell types present in your sample, so that now you can probably have more idea in the blend of fruit juice, what are the fruits that made up this fruit juice. But the problem is still you lose the 3D information. So because in tissue, the cells actually work together side by side, and then they are communicating with each other in the 3D morphology. So when you extract them and then sequence them one in a single cell, you basically lose out this spatial information, and there's also a chance to miss out any rare populations. So, which is why 5 years ago, the trend started to move into spatial biology. So, by doing spatial biology, not only you know the relative abundance of different fruits and what are the fruits that have in your sample, but also how they are spatially arranged in a biological sense for your sample when they collect the tissue section. So for two of these instruments here, the Genomax digital spatial profiler that came out in 2019 is focused more on multicellular level. Basically, you can pick the specific regions on your tissue, and then you can get a more in-depth gene expression, genetic expression or proteomic expression, higher multiplex information instead of just the very small plex, 4 to 5 plex using IHC method. So 2 years ago, they came out with this Cosmics, which is can further enhance the resolution. So instead of multicellular level, you can now go down to single cell and even subcellular level, in which you can see in between the cells, the two cells, for example, the neighboring cells, what are the most differentially expressed gene, and then also the relative or the relative localization. Where are the genes that are most expressed, whether they are in the nucleus, in the cytoplasm, or in the extracellular matrix surrounding the cell. So first we will look at this Genomax digital spatial profiler system first. So when this first system, when we first came out, is basically to help to address heterogeneity of tissue. So for example, if you have tissue section coming from two different patients, one is a responder to a drug treatment, and then one is a non-responder. Then you extract the total RNA from the whole tissue section, and then you do a gene expression profiling. So due to the heterogeneity of the tissue, this most of the time the biopsy will usually contain not only the tumor tissue, but also the surrounding normal tissue. So due to the heterogeneity of the tissue type, chances are you might not be able to find out any significant difference in terms of their genetic expression profile. But what if you are able to focus? You tell the instrument, I only want to focus to profile specifically the tissue that are relevant to your study, for example, the tumor tissue itself, and then just to get specifically the expression profile from that region instead of from the neighboring heterogeneous tissue. So and then in that case, there's a chance for you to pick up any difference in terms of the responder or non-responder group. So if without doing spatial, I think without moving into spatial biology, mainly now the current trade-off is if you do, let's say, NGS or qPCR microarray or even the nCounter platform that I mentioned earlier, basically you just extract total RNA from the whole tissue section. So they are very high plex, especially for NGS. You get a quantitative data and very precise, but you miss out the spatial information because you are profiling everything that you extracted, the whole tissue. Or if you're using a conventional method to do spatial biology, which is imaging. So let's say if you do IHC, you will get the spatial information, but you lose out all the plexing. So mainly for IHC, most of the time you can only go maximum up to 4 or 5 plex, depending on the capability of your microscope. And the data you get is more qualitative instead of quantitative, so the precision is not there. So what this instrument came out with is to address the challenge you have from both conventional methods. So by selecting different regions here, so not only you get the IHC image that you usually see, and then for each of the regions that you selected here, you can get a high multiplex information for either gene expression or protein expression. So in terms of workflow, it's not very different, not much different in terms of for sample preparation, in terms of either you're doing IHC-ish or the nCounter Genomax assay. So similarly, you have this imaging reagent to stain the tissue for you to have the IHC image of the slide to help guide you to choose a specific region you want to have a more in-depth multiplex expression profile. So other than imaging reagent, for nanostring assay, they also include this profiling reagent. So together with these two reagents, you just stain your tissue, and then it's ready to go on the instrument. So here we have a better look on the workflow and then also the chemistry, how it works. So there are two types of different assays. You can do either proteomics or gene expression. So for proteomics, it's basically a target antibody specific to the target proteins in the panel, and then they conjugated it with a oligo that we will use for barcoding, basically used for the instrument during the counting to identify where this barcode was collected from which region on the slide. So they include this photoclearable linker, which I later will explain what is it for. So for RNA reagents, then they'll basically design an oligo, which is complement to the target RNA on the sample. Similarly, it also has a photocurable linker and then is linked to a oligo, which will be used as a barcode for identification. So basically, you add this, you incubate it on the tissue, and then stain them under the microscope on the instrument. So from there, from imaging reagents, you'll be able to get the ISC image. So from there, you can actually pick, so based on the morphological marker of different cell types on the tissue, you can actually pick different regions, probably based on the abundance of different cell types, and then to compare, is there any difference, what is the difference in terms of the expression profile in between the different regions on the same tissue section itself. So for each of the regions that you pick here, the instrument will shine a UV light specifically on the region that you draw. So if you remember earlier, we have this UV photocurable linker probes. So if the shine of the UV light from that region, the linker will be cleaved, releasing the DSP barcode onto the solution. The instrument will now then collect these released photocleave oligos and then aspirate them onto a 96 well plate. So for one slide, basically, when you run, you can technically select up to 96 different regions of interest. So they just basically just collect, dispense onto one well, repeat again for the next region of interest, then dispense it on the next well. So after that, you will collect all these collect all these photocleave oligo and then count it on either using nCounter, Nanostring nCounter platform, or using an Illumina NGS sequencer, and then put back into the count data into this instrument for counting to get the multiplex expression, quantitative expression value for either proteins or genes, depending on the assay they will run. So here are some of the different methods that the user can draw their region of interest. So on the slide, you can either draw something very straightforward, either draw something like a circle or square, so depending on the shape that you like, something straightforward. Or you can do something like what we call segmentation, in which you can actually draw the region following the shape of the tissue morphology based on the ISC image you see on the instrument. So for example, you can select one region for the tumor tissue, and then probably another region as the tumor microenvironment directly surrounding the tumor, and then you can see what the difference in terms of the expression profile. Or you can do something like contour plot, in which you can select one region for the tumor tissue, and then one region that's directly beside the tumor, and then as you move further away, you can draw different regions and then to see whether there's any changes in terms of their expression profile. Or you can just do something so like random. It's a gridded form. That means it's not biased. There's no selection of the region based on the tissue type. You basically just draw a range gridded form to profile each region. So you can also do that. Okay, here I have a example data. It's a tonsil sample stained using two different markers. So one is on Ki67, which is a cell proliferation marker, and then one is the CD3 immune cell marker. So here they pick 4 regions of this sample data based on the difference in the abundance of different cell types. So one is enriched in Ki67 positive cells, and then one is enriched in CD3 cells, one is a mix of both, and then one is basically negative for both markers. So by running the assay on Genomax platform, other than the qualitative image, what you can get is also a quantitative data, numerative, quantitative expression level, for different analyte, and then you can compare between different regions to find out the significant fold change. And here I have another example data, which basically what they do is they have this single tissue section. They selected 12 different regions based on the different abundance of different cell types, either CD20 enriched, CD3 enriched, pan-CK enriched, or a mix of different cell types. So what they can find is they run this is actually running on a protein assay, and you can see here different regions actually shows a distinct expression profile, protein expression profile. So imagine previously for your conventional method, you have been extracting total RNA from the whole tissue section for profile. Basically, it's a blend of all these signals. So you miss out all this, what is happening in the tissue section itself. Here I have another example. What they have here, they actually stain the cells with RNAscope. So other than antibody reagents for imaging, Nanostring also has option for user to use RNAscope, which is a RNA probe-based imaging reagent to stain for IHC image. So here, this is the staining on the tissue staining. This is the image, and then the user pick the different data from different regions on the slide based on whether they are expressing high in CD274, which is PDR1, either PDR1 high or PDR1 low. And then they do a data analysis, and here is the volcano plot. So you can see here two different regions, PDR1 high or PDR1 low. There are different genes that are differentially expressed in each region. As a proof of concept, Nanostring also has this data in which they profile a pancreatic tissue, and then they selected alpha cells as one, or select all these specifically the islet of Langerhans, and then they select alpha cells as one region and beta cells as one region. So I think you're probably familiar with pancreatic cells, which is alpha cells and beta cells. They are playing a vital role in the glucose regulation of glucose in the blood level, blood sugar level. So you can see here, definitely two the alpha and beta cells are expected to carry out a very distinct or different cell function. So as a proof of concept, when they do a data analysis and then the volcano plot, you can actually see there are a list of genes, a group of different genes that are differentially expressed each cell type. For example, here alpha cells expressing high in glucagon gene, beta cells higher expression of insulin gene. So here we have a look at some of the panels that are offered by Nanostring. First, they came out with this cancer transcriptome atlas, more for cancer researchers. So this is a highly multiplex panel. So it provides over 1,800 different genes simultaneously and with spatial information for each of the region that you selected on the tissue section. So this one covers a very comprehensive content for cancer biology research in general. It includes coverage for global immune response, microenvironment, immune activity, tumor reactivity, and so on. And it covers over 50 pathways to explore more aspects of cancer and also tumor biology. Or if you don't have a specific research focus in mind, they also have this whole transcriptome atlas, which basically look at all the known genes that in the transcriptome. So either there are two panels, one for human, one for mouse. So for humans, it can profile up to over 18,000 genes, and then on mouse, it's even more, over 21,000 genes. So with spatial resolution for each of the region that you selected on the tissue section. So if you do not want to get a large data working with full transcriptome, you can also opt for a more specialized smaller panels. For example, here, panel looking at just the immune pathways. Or if you're working with canine samples, they have also a panel for that canine cancer atlas. Or if you're not looking at gene expression, you want to do protein assay, they have also panels for that. They have two different panels, one for cancer immunology and then one for neuroscience. So from there you can pick up to 96 proteins from the more than 140 targets available. And more recently, they also came out with a much larger proteomic protein expression panel, over 570 plex, which is called the IO Proteome Atlas. IO stands for immuno-oncology. So here are the group of proteins that are included in the panel. So the protein list can be divided into 77 annotated pathways across 556 genes, and also include coverage of more than 60 phosphorylated targets. So although the name of the panel is called IO immuno-oncology, but other than immuno-oncology research, can also be used for other research areas as well. For example, autoimmunity, infectious disease, and organ transplant. So most of the time, it's always good to. What we usually recommend is to share. We will share the protein list with you, and then you can have a look at all the protein list, and then see whether this is something that is suitable for your research focus. So for Genomax, I think one of the main differentiator compared to the other platform on the market would be how they can, in terms of is the flexibility it gives the user to draw the region of interest on the tissue. So you can specifically pick a specific region based on the ISC image to pick the specific region you want to have the expression, a more in-depth expression profile. So you can either draw a circle, you can also follow the shape, the morphology of the shape of the tissue. So compared to some other method on the some other platform on the market, probably they will only use a dot matrix in which they just collect the information for only the specific fixed region on the tissue. So chances are you will still get some useful data from here due to different region covering different spots covering different region on the tissue, different cell type, but it loses the flexibility there. So you also miss out anything what is happening in between all the empty spaces here. So Nanostring gives you the flexibility to pick specifically the region that you want to profile on the tissue section itself. So for Genomax, it works with all types of tissue samples. So technically, as long as you can fix the tissue onto a glass slide, it can be profiled on Genomax. So like core needle biopsy, FFT tissue block, slide mounted tissue, fresh frozen tissue, you can also work with the platform. So including TMA, tissue microarray sample type as well. So sometimes what other people do is they can actually, what you can do is you can actually have multiple core, one core coming from one tissue section. So technically you can have multiple tissue sections fixed onto a slide by doing a tissue microarray sample type. So on a single slide itself, it obtain information, expression information from different patient or different tissue section. Okay, so now we can move on to the next platform, which is called the CosMx Spatial Molecular Imager. So currently, this instrument is able to profile up to 6,000 plex for gene expression, and then they are now also try to increase the panel size. So by next year, they actually announced that they're going to come up with I think it's 11,000 whole transcriptome atlas panel for user. So with that, you can actually get a very high plex multiplexing information down to a single cell or even subcellular level. So similar to Genomax, CosMx also works with multiple types of tissue sample, FFPE, fresh frozen, tumor microarray, or even organoid sample. So it's also a multi-omic platform. You can profile either RNA for gene expression or also for protein expression. So it provides high resolution as a data, so you can go down to subcellular level, providing you a three-dimensional resolution. So here's just a quick look how your data will look like if you look at different levels. So for whole tissue section, it's a bit similar to what you see on Genomax earlier. So you can see overall on the whole tissue section, different regions, what are the genes that are most abundantly expressed, and also the cell. If you all their panel also include the cell identification, cell typing probes, so you can actually also find out what are the different cell types that are present on the tissue section. But other than that, on CosMx, it can go beyond, in which you can actually zoom down to single cell level and then see for this specific cell what are the most abundantly expressed transcript, gene or protein, and then where are the relative expression location. So if you go down to the subcellular level, you can actually see the transcripts, whether they are expressed mainly in the nucleus, the cytoplasm, or in the tumor, in the ECM extracellular matrix. So here's just a graphical representation of their data, how they can take image. So not only they take the detect the signal on X and Y axis, but they also detect on the Z axis, which you can actually get in a way like a 3D, three-dimensional out view of your data. So for cosmics, they uses what we call in situ hybridization chemistry. That means the probe will directly bind to the target on the tissue section, and then the detection itself is also happening on the tissue section itself. That's why it's called in situ hybridization chemistry. So they use a cyclic imaging method. So if we share, it's a bit technical, but basically I'll just give you a quick brief through. So what they have on this probe is a readout domain with 4 different regions here that can be bind by this reporter probe. So this reporter probe will have 4 different fluorescent colors, either blue, yellow, green, or red. So depending on the sequence there's contained on the readout domain here. So different target, different probe targeting different gene or different proteins will have a difference in the sequence of the real domain. So different probe here, we have different reporter colors bind to it. So different color combination can help the software or the instrument to identify for this signal that we detected which gene or which protein this came from. So other than protein expression, RNA expression information, and you can also get cell typing information. So for the cells segmentation carried out by the instrument, they actually combine 2 different models. So one is a nuclear model, one is a cytoplasmic model. So basically combine these 2 different models here to better segment the cells accurately, the individual cells here, so that the information you get from the individual cells is more accurate. There's no overlapping between different cells in terms of the expression profile that you get, the data that you get. So here, this is we're just looking at the 1000-plex panel. So although they have now even came out with a larger panel at 6000-plex, but for their 1000-plex panel, they not only include the markers for cell typing, but also for markers to tell the cell state function. So what are the state of the cells when the tissue has been collected from the patient, and then what are the functions that the cell is currently it was carrying during the collection of the tissue, and then also markers for cell-to-cell interaction or hormone activities. Okay, some of the sample data coming from different tissue types, either FFPE, formalin fixed, paraffin embedded tissue, or even fresh frozen tissue, tissue microarray organoid. So it basically works with, as I mentioned earlier, multiple different tissue types. So here's the quick view on the workflow. Basically, you have to, for FFP sample, you have to immobilize them, fix, and then retrieve the target, and then incubate them with the probes, RNA or antibody probes, depending on which assay they run. And then you assemble it, put into the instrument for the read. And then, because it's the in situ detection, so there's no probe collection or like Genomax. This is directly in detection inside on the slide, on the tissue section itself. And after that, you can do data analysis, either offline using the platform, using the instrument itself, or they have the solution for online analysis using the Atomics platform. So this Cosmics currently, this is the slide is actually a little bit old, so I believe now even more customer projects has been delivered for a lot of institutes around the world. And more than 15 tissue types, either normal or disease, for human or mouse, has already been profiled for Cosmics as well. So for example, brain tissue, lung tissue, kidney, colorectal, liver, pancreatic tissue, all has been tested and proven to be work on this platform. So the system is able to provide high sensitivity, yes, wide dynamic range to be able to detect low expressors, medium, and also high expressors. And you can detect more than thousands of transcripts in a single cell. So if you have been working with NGS, you're probably familiar with this Disney plot, in which based on the data they collected from the tissue section, the the plot here can actually help to identify and also isolate out what are the different abundance of different tissue cell types that are present in the sample. But actually, this is technically it's not a 3D or actual reflection of the distribution of the different cell type of the tissue. It basically just tell you there are different cell types, and then how different are they in terms of the expression profile. But if you run on Cosmics, then you do get the signal, and then to be able to identify the different cell types, and then where are they distributed on different region on the tissue. So you have an example here, different cell types, they are distributed at different region, and then different tissue type also have different cell type might also have different abundance on the tissue. So as I mentioned earlier, this platform doesn't actually just only give you the cell typing. So it doesn't only give you the cell type to identify the individual cells, but they also include probes for cell state, cell function, to actually give you some insight on what is happening to the cells, what is the cell trying to do, or the function is trying to carry out during when the tissue has been collected. So for example, here is zooming into these microglia cells with the neighboring interneuron cell. Then with the abundance of the expression of microglia markers, it suggests that these microglia cells exhibiting a mildly phagocytic phenotype, also based on some blue dots here, which is the expression of the phagocytic marker. So without cell state or without cell function, cell state information, if you only run cell type assay, then basically this is the only information you'll get. So these are microglia cells, and then neighboring them, there are different cells. That's it. But with a larger panel that includes cell state, cell function probes, you can now also tell at that moment what is the cell trying to do. So the microglial cells, for example, here you can see here 3 different regions on the tissue. The cells actually express different level of phagocytic marker indicated by the blue dots. For example, here, the first and second picture here, they are only expressing a mildly phagocytic markers compared to the third one, which has a much higher expression level of phagocytic marker compared to the other 2 microglia cells at another region. So this suggests that these 2 microglia cells specifically here, they are trying to carry out phagocytosis. So here are some of the panels that are currently offered by Nanostring of their catalog. So they have RNA panel, protein panel for both human and mouse. So depending on your research focus, they have also specialized panel for that, for immuno-oncology and also for neuroscience. So eventually, they also came out with a new panel. It's a 6K discovery panel. So here are the coverage of the genes they are included in the panel here. So they are looking at a lot of different cell functions, different signaling pathways to give you further insight on your spatial data. So just to give an overview of 2 the different platforms. So depending on your research focus, so sometimes probably multicellular level is already good for you. In that case, then probably genomics is already sufficient. So for example, you just want to profile different different region on the tissue section to find out, or you just want to specialize on the tumor tissue region, for example, to profile and then compare between different tissue section or different patients, control versus disease, for example, to see what are the difference, the gene expression profile or protein expression profile. You do not need a single cell resolution to help resolve the heterogeneity of the tissue. Then genomics is suitable for you. But if you need to look into single cell level to want to find out for each of or if the population that you are looking at is a very rare population, then or they are like scarcely spread out throughout the tissue. There's only 1 or 2 cells at a time for each of the region they profile, then probably CosMx will be more suitable for you. You also want to go down to single cell level or even subcellular level to see each of these cells here, and then based on the region they are located on the tissue section, probably they are carrying out different function or at different state. If you need those kind of information, then CosMx will be probably more suitable for you. Okay, so before I wrap up my talk for today, I'll just have a quick walkthrough on the nCounter platform, which came out much long before these 2 platforms that I introduced earlier. So this is the Nanostring nCounter platform. They have currently 3 models on the market. So they actually have panels to run either for RUO or for the diagnostic purpose. So currently on diagnostic, they have one panel, which is called the Prosigna Breast Cancer Assay Panel, which can help to do a breast cancer subtyping, and then also provide this prognostic score, the risk of recurrence score, to give information on for this patient if they are to undergo only a adjuvant hormone therapy, what will be the risk of recurrence of the tissue of the breast cancer. So what's the risk of it will come back in the next 10 years? So this is their diagnostic panel. So for their research use panel, currently there are 2 different models, 2 different instruments that can actually work with their research use panels. And currently, actually, in Klang Valley area, we do have each of these units here. We have 2 labs. Each we have a different unit, different model that can actually allow the external user to use the instrument by paying just a small amount of access fee. So that means it's actually possible for our customers to run the nCounter assay on the instrument without buying the instrument itself. You just need to purchase a kit from us, pay the access fee to the lab, and you can run the Nanostring assay. So for their research use panels, they actually have a wide range of panels for different research area for their gene expression panel. And they have also panels for microRNA expression, gene fusions, and they have 1 panel for DNA assay, which is the copy number variation. So here we have a quick look on how the assay works. So it basically works using what we call a molecular barcode, and it works with a 2-probe hybridization system. So you have this capture probe and reporter probe. The capture probe is bioattenuated, and then reporter probe is conjugated with this 6 fluorophores, which is used as a barcode, a molecular barcode. So how it works is there's 4 different colors, and then the probes, the dyes are arranged in different position. For example, here different color combination can actually help the instrument to identify different genes that's present in the sample. So this is a bit similar to when you go to the supermarket to do grocery shopping. So when you're trying to pay at the cashier, what they need to do is just to scan the barcode on the item, and then they will know automatically what are the items in the shopping cart and then what's the price. So this is similar to this Nanostring. So they use this molecular barcode to identify the genes that is present in the sample. So each of the probe here, there are these 50 base target which will bind complementary to your target sequence. So when combined, the target sequence they bind is 100 base pair. So one of the main difference for Nanostring nCounter platform compared to the other conventional platform to do gene expression studies like NGS, microarray, or qPCR is that Nanostring nCounter directly works with total RNA. So basically, just what you need to do is just extract your total RNA from your either tissue or cell sample, and then do nano drop reading to find out the concentration and also the 260, 230, 260, 280 ratios. If they pass the requirement, they can be directly used on Nanostring. So there is no need to do cDNA synthesis to convert to DNA. There's no need to do amplification for the detection. So it works directly with total RNA. So there's no enzymatic reaction. So for this reason, actually, Nanostring nCounter platform works very well with degraded samples like FFPE sample as well. And then also, if you have sample contamination carry over from your extraction process, usually they are also less predictable compared to qPCR. So this is just a quick overview on the whole workflow of an nCounter. So the only hands-on part is actually just the first step, in which you have to create a mix of the probes, and then together with your total RNA, and then you just put into a thermocycler for overnight incubation for the hybridization process to occur. On the second day itself, usually everything is automated. So you just need to load your sample to the instrument. The instrument will do the washing steps and then also the scanning step. So another difference between Nanostring nCounter with let's say qPCR or microarray is when you measure the fluorescence, it doesn't really measure the fluorescence intensity. So it doesn't quantify the expression level by fluorescence intensity. Instead, it measure the fluorescence signal just to identify which barcode is this, then which barcode this gene identify to. So for each of the barcode they counted here is actually equivalent to 1 molecule of messenger RNA if you're running gene expression assay. That is because each of the barcode that you see here is actually coming from one set of probe hybridized with 1 molecule of messenger RNA. So this method is what they call a direct counting. So it directly count the number of molecules that is found on the scanning area on the cartridge, which is a bit similar to NGS. NGS is also considered as a digital counting platform because it after it does a sequence alignment, it can actually count the number of transcripts detected for each of the different genes in the sample. So when you compare the fold change, is actually fold change between different samples is actually the difference in terms of the number of transcripts detected in each sample. So the fold change value, I would say, is probably more numeratively accurate and then more meaningful compared to if you're using, let's say, qPCR, basically a difference in the CT value, which is the CT value is called when the signal has reached a certain threshold. So in a way, there is a bit of an approximation because of the quantification. So same goes to microarray. They also try to, when you stain your sample with 2 different probes, they want to find out which probe has a higher signal intensity, then that one has the higher expression level. So it's also more qualitative in a way compared to nCounter. So on the instrument, this will be what you see under the microscope when they try to detect the signal. But for end user side, it's actually very straightforward. What we get is directly the raw counts for each of the genes. And then usually their panel will also include the housekeeping genes for you to do housekeeping gene normalization. So after the normalization, then you can directly compare the fold change between different sample groups that you included in the run. So another advantage of nSolver is for Nanostring, for Nanostring, they actually already have the data analysis software for free, which is called the nSolver. So you do not actually need to pay extra just to get the data analysis done. So on nSolver analysis, nSolver software, you can do some basic analysis like heat map clustering, box plot, violin plot, scatter plot, or if you use their off-the-shelf panel, due to the gene list is already well annotated in their panel, you can actually also do some other further analysis. For example, like pathway scoring to find out for not just the individual genes that are differentially expressed, but as a group of genes, do they show a trend in between different sample groups? For example, here, a group of genes that are involved in apoptosis. And then when they compare to different sample groups here, let's say, for example, here, it shows a decrease in the score from sample A to C, indicating a loss of function or cells are no longer carrying out the apoptosis function when you move down from sample A to sample C. And they can also plot a volcano plot to tell you when you compare between 2 different sample groups, what are the genes that are differentially expressed, whether they are up or down regulated, and then whether they are significant in terms of their adjusted p-value. So if you run a panel that is more specialized on looking at genes involved in various signaling pathways, the software can also help to do what we call path view analysis, in which they pull out the plot from KGT database, and then they do a color coding between you compare between different sample groups. So if they have a significant fold change, they will be color coded. So orange indicates high up regulation, blue indicates low regulation, gray color there's no significant fold change. The white color box basically means the genes is not included in the panel. So other than that, Nanostring also collaborated with third-party data analysis bioinformatics platform for the on-ramp bioinformatics. So they have a product called Rosalind. So this is a cloud-based data analysis platform, which is of due to the collaboration, they allow nCounter user to analyze their data for free using the platform for a year. So basically, there's two different options for the user, the analysis pipeline for the user to try out. So Rosalind is more like a more interactive platform, which I usually also like to recommend our user to try it out, because it's highly interactive, and then they also have more information for more database. So sometimes it might be able to give further insight that might be missed out by the nSolver analysis software. So just to summarize the advantages of nCounter technology. So as I mentioned earlier, there's no amplification. It works directly with total RNA that you extracted from your sample. So for that reason, the sample input is very flexible. It's very tolerant to degraded sample like FFPE, and also more tolerant to sample contamination, for example, carry over from extraction process compared to other platforms. And it do direct counting, digital counting, and due to the highly automated process of the whole workflow, it has a very high reproducibility of nearly 6 logs of dynamic range. It's very highly multiplexed, so it can provide up to 800 different targets for a single reaction tube that you set up. Workflow is also very straightforward. So technically, it's just only 4 pipetting steps. Basically, just to create a mix of the probe, electrode to a sample tube, put in a total RNA, lastly, put in the capture probe to each well, and then put in the thermocycler for overnight incubation. Everything else on the next day is just automated. So the whole flow will just take around 1.5 days. So you can start on the first day afternoon. By the late afternoon of second day, you can already get your data. So there's no library prep like run NGS. There's also much less pipetting steps compared to qPCR. So you have much lesser human error or pipetting error. So in the last part of my talk, I'll just quickly brief you through some of the panels that are currently offered by Nanostring for their gene expression panel offered by Nanostring on their catalog. So these panels, they are all usually carefully curated by their scientists. So based on the publications, which are the genes that are reported to be clinically relevant, so all of the genes will be included in the panel. So one of the most hot-selling panel, actually not only around the world, but also in Malaysia, is their pan-cancer panel. So there are 3 different panels to address different research needs. Each of them are 770 plex. That means when you run one panel, you can get expression profile of 770 different genes from a single sample. So there are panels focusing on the 12 canonical pathways, or they have also panel looking at how the immune system of the patient respond to the cancer, and also how the cancer progress over time in the patient. So they have also other oncology panel looking at either immuno-oncology, breast cancer, tumor signaling. If you're looking at more on the metabolic pathway, they have also a panel for that. Antibody drug conjugate development panel, if you're doing relevant research, T-cell receptor diversity, and if you're working with canine animals, they have also a panel for that, canine immuno-oncology panel. So other than oncology, Nanostring also has a wide range of panels for immuno-oncology research. So for example, here they have a general immunology panel or information panel. If you are more focused on studying autoimmune disease, they have also a panel for that. Studying fibrosis, immune exhaustion, immunity, they have also a panel for that. And other than human or mouse, they have also a panel for non-human primates immuno-oncology study. So other than that, they have also panels to cover different research areas like infectious disease research. They have this post-response panel that actually came out just 2 or 3 years ago during the COVID-19 pandemic. So because during that time, the research is very much focused on infectious disease, specifically COVID-19. Then they came out with this panel that can actually help to study the host, the 5 phases of host from the initial infection phase down to the peak infection and then the recovery phase, see what the genes that are differentially expressed. If you're doing CGT, cell gene therapy research, they have also a panel for gene therapy optimization, CAR-T, cell characterization, stem cell characterization as well. Or if you're doing neuroscience research, they have also panels for neuropathology, neuroinflammation, Alzheimer's disease as well. So most of their panels, all the panels that I've been through earlier, they are all mainly more for human or mouse. But let's say if you are working on some other organisms like plants or other organisms like zebrafish, or you are doing veterinary medicine, studying dogs, chicken, cows, for example, then you can do process customization. Basically, what you need to do is just to share the sequence or the RefSeq ID. If the sequence has already been published, just need to share this information, a list of target genes that you want to study to the bioinformatics in Nanostring, then they can help you to design the probes to specifically target the genes that you're trying to study. So yeah, that's all for my sharing session this morning. So thank you for your time.